For the invitation and for that great introduction. You sure you're talking about me? Yeah. And thanks you all for coming, and I look forward to, I hope, try to make the talk interesting for you, and I hope that it leads to some interesting questions at the end. So this is Sherman's Lagoons, the only cartoon series I know of about marine biology. So you can access it. There's a little cartoon every day. It was worth looking at. Some of them are pretty funny. And so it's an acidifying ocean. Oh, how do I advance the slides? There. So an acidifying ocean, where might it lead? That's what I'll be talking about today. Here's an interesting graph. This is really the source of most of our global issues in the world today. So 10,000 years ago, there were hardly any people walking around, and that was true for many thousands of years. And then around year 2000, the population growth really picks up quite a bit. And, 2000 BC, 2000 BC. And then as we get closer to our own time, this is really quite amazing. It just goes straight up. And that red part of the line really coincides with some number of interesting inventions. So the invention of vaccines, antibiotics about 70 years ago, the Green Revolution, so food supplies are much more available to people. When I was born, there were about 2.5 billion people on the planet, 2.5 billion. And now there are about 3 times as many people on the planet, about 7.4 billion last I counted. And the other thing is that more and more people want to live like we live. They want to be able to drive their big cars everywhere, have bigger houses, heat them well in the winter, cool them off with air conditioning in the summer, and eat good food: chicken, steak, salmon, And so there's some problems that are being created. One of which is increased destruction of natural resources, including forests, and also increased consumption. You have more people eating more food, and they're eating higher up on the food web. So here's another graph showing, this is starting from about 1955, and the gray line, again, shows human population growth on the x-axis to the left there. So we've seen that before. The dotted line up above that shows world fisheries production in tons. Okay. And you'll see from 1960 to about 1985, there's been it was quite a large increase in fish production around the world, mostly due to technological advances. So you can now, with sonar, underwater sonar, you can find fish schools really easily. And we have the technology now. You can pretty much catch all the fish in the school. So our fishing efficiency has gone up quite a bit. But since about 1985 or so, the fish catch has not increased, and for many fish populations, it has plummeted really seriously. Human population size keeps increasing, but the fish catch has not kept up, and it's diminishing. So that's interesting. So increased consumption is a problem. Destruction of natural resources is a problem. How you can more and more people, more consumption, more waste production, and then, of course, increased pollution, including the excess carbon dioxide emissions that I'll be talking about today. Okay. So this is monitoring carbon dioxide in the air, a little place off the coast of off in Hawaii. So away from big cities, away from a lot of transportation, so you don't have a lot of exhaust being created on the island. And so the data are really pretty accurate estimates of what's actually in the air. And you see about 1960, there were about 315 parts per million of carbon dioxide in the air. And it goes up quite a bit from there. And in fact, now, just I think last year, we went over the 400 parts per million line. I think it's the highest carbon dioxide levels in the atmosphere for about 400,000 years. The last estimate that was a long time ago. That was before I was born. Yeah. And here's another graph. So the red line at the top, again, is the atmospheric oxygen concentration, going up from about 350 to 390, 380 something. And below that is the carbon dioxide in seawater, surface seawater measurements. There's a lot of fluctuation there, but it clearly going, the trend is upwards pretty clearly. And down below here we have the pH of the ocean water starting at about 8.1 and going down by 8.09 a little bit now. In a lot of places it's about 8.0. Okay, so it's dropped. So the pH is a weird scale, and somebody should apologize for creating this because it caused a lot of confusion. But the idea is as acidity of the water goes up, the pH goes down. So lower pH corresponds to higher acidity. What is acidity? In fact, ocean acidification. What's acidity? Anybody? Concentration of hydrogen ions. Yes, concentration of hydrogen ions. So you're dissolving CO2 in water. There's no hydrogen in CO2. CO2, it's carbon and oxygen. So where are these hydrogen ions coming from? There they are. So you have carbon dioxide dissolved in water. You got carbonic acid. The carbonic acid dissociates to form bicarbonate ions. There they are. And hydrogen ions. And the bicarbonate ions can further dissociate to form more hydrogen ions, release more hydrogen ions, and carbonate ions. So you get there's the result of carbonic acid formation that eventually gives you these hydrogen ions. Okay. From what I've just told you, actually, how could you use this information to construct an argument proving that global warming must be occurring? Carbon dioxide is a greenhouse gas. What does that mean? So sunlight comes in, warms up the land, warms up water. That warmth radiates. You're radiating heat from the water and the heated up land. But those wavelengths, so sunlight gets through our atmosphere pretty nicely, but the wavelengths that are radiated by heated up surfaces are trapped by greenhouse gases. That's why they're called greenhouse gases. So carbon dioxide is one of those gases. Methane is another important one. Carbon dioxide is another one of those gases. So if the ocean is getting more acidic, which it certainly is, then that must prove that there's excess carbon dioxide in the atmosphere. And if there's excess carbon dioxide in the atmosphere and it's a greenhouse gas, the planet must be warming. Does that make sense? How can you argue against that? I don't know. So spread that around. See if anybody, they may just drop their jaws or something. I never thought of it that way, but it's happening, and it's hard to. If the ocean is becoming more acidic, then the planet must also be warming because carbon dioxide is a greenhouse gas. And about a third of the excess carbon dioxide is absorbed by the ocean. Okay, so that's a good thing in a way, because if that weren't happening, the planet would be warming up even faster. Well, a lot of biologists have accepted a lot of grant money over the years to research the effects of ocean acidification, and chemists have certainly gotten involved too. So here's something that's worth reading. The anthropogenic carbon emissions caused by the burning of fossil fuels, cement production. Cement production, it turns out, the chemical reactions forming cement release a lot of carbon dioxide. I think I've seen estimates that maybe 5% of all the total excess CO2 emissions are from cement manufacture. So how much carbon dioxide are we going to release if we build that wall? A lot. And deforestation, of course, is the problem, having a major impact on the world's largest ecosystem, the oceans. Atmospheric carbon dioxide is the highest it has been for at least the last 15 million years. And probably longer. That's a long time. The geological record suggests that the current acidification is potentially unparalleled in at least the last 300 million years of Earth history, and raises the possibility that we are entering an unknown territory of marine ecosystem change. Okay. This is a paper to an Introduction to an Ocean Acidification issue in a journal in 2016, and Howard Brauman wrote about how many people have been working on ocean acidification over the recent years. So it's a change in seawater chemistry driven by the uptake of atmospheric CO2 by the oceans. It's probably been the most studied single topic in marine science in recent times. According to the Web of Science, no articles used the term ocean acidification before the year 2000. None. 5 articles responded to the search term OA in 2005, although only 2 of those dealt with the topic in a manner consistent with contemporary research. From 2006 to 2015, 3,100 articles on ocean acidification appeared. It's 300 per year. In 2015 alone, more than 600 articles were published on this topic. And at the time of this writing, one of the first articles on this topic had been cited more than 1,500 times, according to these people that track these things. And just about an hour or so ago, I looked online to see how many papers have been published on ocean acidification since the start of 2017, and there are almost 2,000 papers that have been published so far this year on ocean acidification. I tried keeping up with that literature. Okay. So let's take a look at some of these papers and see the sorts of things that people have been looking at. The ocean acidification causes bleaching and productivity loss in coral reef builders. Coral reefs get a lot of press because they're really pretty, and they're also one of the most complex ecosystems on the planet. So ocean acidification causes bleaching. So what's the bleaching come from? Turns out that coral reefs live in what we normally think of as biological deserts. You know, the reefs are so dynamic and they're so diverse. It's hard to imagine that they're actually surrounded by what's correctly termed a biological desert. The problem is, in the tropics, the water is always warm, and it's very low in nutrients because the nutrients are released when things decompose in deeper water. And the deeper water is cold. So there's a thermal barrier, a density barrier between the upper warm water where you get all the sunlight and the colder high nutrient water. And it's like a wall. It's like a wall. And so the nutrients have no way of getting up into the surface waters. And so that's why the water is so clear. Nothing's growing in it. The corals have developed an amazing symbiotic interaction with single-celled algae that they actually incorporate into their tissues. And so now we have plenty of sunlight. The algae photosynthesize like crazy. They pass on the carbohydrates they make to the corals. And where do the algae get their nitrogen? Basically, the corals pee, and they get the nitrogen from the corals' waste production. So it's a nice little symbiotic relationship, and that is the entire basis for this amazing ecosystem that we have in the tropics. So the bleaching, ocean acidification causes bleaching. The problem is that the symbiotic algae are either killed or they abandon the coral, and then the coral has no food source, and the corals die. And this is what you get. And apparently, if you snorkel, it's only like a couple feet of water, so you don't even need a scuba tank to see this stuff if you go to the right place. And so you can go for square miles and see nothing but this in major parts of the reefs around the world now. Projected near future levels of temperature and partial pressure of CO2 in water, so more CO2 is more acidity, reduce coral fertilization success. So corals are animals, just like us. People always talk about people versus animals. It's really annoying because people are animals. And corals are animals too. They make sperm, they make eggs, and the sperm fertilize the eggs, and they spawn in huge numbers, and it's very coordinated. So they may look stupid, but somehow they coordinate their spawning. I mean, if you release your eggs one week and then the males release their sperm a couple days later, it's not going to work. So it's very coordinated, and you get huge numbers of gametes in the water at certain times a year. Okay, and the eggs get fertilized, and they develop into little microscopic larval stages that then swim around for a number of hours or days before they attach to a surface, a rock surface, and start turning into corals. Okay. Yeah. So reduced coral fertilization success, that could be a problem. If you don't have fertilized eggs, you don't get larvae and you don't get new corals developing. There's a lot of work that's been done also on blue mussels, Middleus edulis, which are very common along the coast of rocky intertidal zones from Maine down to, I don't know, pretty far south along our coast. So effects of increased seawater, seawater should be one word. Okay. Effects of increased seawater concentrations of carbon dioxide on growth of the bivalve Middleus edulis. So here's some bivalves in the field at low tide. You see them in really large concentrations. And there's an effect on the growth rate. So the shells tend to grow more slowly. They also tend to be thinner. And if they're thinner, they're more vulnerable to attack by predators. So that's one study. It turns out that mussels, actually all invertebrates, have non-self recognition systems. They're not as sophisticated as ours, but they exist, and they play important roles in the health of these organisms. And it turns out that ocean acidification not only affects shell growth, but also affects the immune responsive capabilities of this particular mussel. So that's probably not good. They're going to be more susceptible to diseases. This is an interesting one. There's a mussel, and with these threads sticking out. So the mussels have a foot, and the foot secretes, in a very complicated way, it turns out, these beautiful little threads, which have little sticky pads at the ends, and they're glued to a rock surface. So they glue these strings one at a time to a rock surface, and then build the rest of the string. And the strings are very elastic, so there's a lot of interest in this process, and in terms of as for surgical, particularly for human surgery applications. So people are starting to look at maybe in heart surgery, open heart surgery, you can use this glue to seal up the wound. It's flexible, it hardens underwater. Anyway, so the mussels, they look stupid, but they actually come up with some pretty interesting things that turn out to be useful for us. Anyway, so they secrete these threads, and this anchors the mussel to the hard rock substrate that they're trying to stay with. Which means when waves, the tide comes back in and they're subject to waves, the mussels aren't washed away by the wave action, and these threads are very elastic, very, very elastic. So the wave comes in, and the threads stretch, and then they go back to normal, and so that the mussel is able to withstand incredible wave action without being washed away. And also predators that eat the mussels usually have to pull, like a crab, for example, they have to pull the crab or pull the mussel off the substrate in order to get at it and crack the shell. And so here's one that's really pinned down. He's got a lot of byssal threads there. It's going to be pretty hard to remove that animal from the substrate it's on. So if this byssal attachment is weakened by ocean acidification, it's going to make it easier for waves to wash the animals away, and it's going to make it easier for predators to pull the animals off and eat them. Why is there so much work on blue mussels? Aquaculture is really growing. Blue mussels are a major aquaculture animal. They're pretty darn tasty, I have to say. Yeah. So a lot of interest in corals because there's some interesting financial impacts. Okay. Fair amount of research on oysters for a similar reason. People are interested in oysters and becoming very major importance in aquaculture. So interactive effects of salinity and elevated carbon dioxide levels. People are starting to do more studies where there's several environmental factors varying. Salinity is one of them, temperature is another one, pollutants. People are just starting to do those multi-stress studies. So interactive effects of salinity and elevated CO2 levels on juvenile eastern oysters, and the effects are increased mortality, slower growth of the oysters, thinner shelled oysters. And so if you're growing more slowly, you're smaller for a longer period of time, and your shells are thinner, it's going to be easier for predators to eat you. So it's bad for the oyster. Could be good for the predators, though, which is interesting. Oh, problem. It turns out that ocean acidification has some really unanticipated effects on behavior. I'll say more about that in a minute. But ocean acidification, it turns out, interferes with the ability of some crabs to forage effectively, and they actually did the study on oysters. I stuck the on oysters in there. That would have been a good thing if you were writing this paper to put that in the title. Anyway, so the oysters are smaller and their shells are thinner, but the crabs that prey on them have more trouble finding them and more trouble handling them for some reason under acidified conditions. So what's the consequence of that? Hard to say. And here's another study, unshelled abalone and corrupted urchins. Development of marine calcifiers in a changing ocean. So again, a lot of work. Abalone is another commercially important species on the West Coast, very, very important, a lot of money tied up there. So the West Coast people are very concerned about, and they're already seeing effects on their aquaculture. Aquaculture is having more and more problems out on the West Coast in particular because the pH problem is even worse out there than it is on our coast. And sea urchins have some commercial value too because the sea urchin gonads are pretty valuable. So again, a lot of the research is focused on things that have some commercial importance. Okay. And pretty much anything that forms a calcium carbonate shell, people are looking at. And there are worms that are a lot more interesting than earthworms, for example, though they were getting interesting enough for Darwin to write. Actually, his last book was about the behavior of earthworms. Anyway, so marine worms are pretty diverse, and they do a lot of things that earthworms don't do. And one of the things, they make these calcium carbonate tubes, pretty conspicuous, that are attached to rock surfaces. And here's one tube with little worms sticking its forehead out there, and they filter food out of the water as it comes by. And as they grow, they just elongate their tubes. And CO2-driven ocean acidification alters and weakens the integrity of these tubes. Okay? And so again, it's going to make them more vulnerable to predators. So there's a lot of work on the calcification process and lots of different aspects of it. But it's not just those organisms that have been looked at. So here's some studies. Here's a study that actually focused on the effects of ocean acidification on copepods. And copepods, how many people know what copepods are? How many people have ever seen a copepod? There you go. They are probably the most abundant animal on this planet. They are really small. They're really small. They're less than a millimeter, typically half a millimeter, and but they're extremely abundant. And there are many, many species. If you take a plankton tow off the coast here, over the course of a year, you'll encounter something like 350 species of copepods in the local waters. And I guarantee, every time you go to the beach, if you swallow a mouthful of seawater, you are swallowing dozens of copepods at a time, at least. So I usually tell my students when they go to the beach in the summer to bring along some garlic butter. Yeah. They're really important because they are the basis for pretty much all marine food chains, all major marine food chains. So these things eat single-celled algae called phytoplankton. I'll say more about those in a minute. And they then serve as food sources for many other organisms, including the larval stages of a lot of commercially important fish. So copepod populations diminish. That's a problem for the fisheries. So on the right there's a female copepod. It's got two little egg sacs she's carrying around with her, and she'll be releasing those soon. There'll be lots of little larval copepods swimming around. Yeah. So it turns out there's all kinds of effects. The ocean acidification conditions reduce the fecundity, the number of eggs produced by the copepods. They reduce the survival of the developmental stages. They reduce the growth rates of the animals. There are just lots of effects. There's the ocean acidification disrupts induced defenses of intertidal gastropods. So this is common periwinkle. So the tide goes out at a rocky area near you. You'll see a lot of these periwinkles on the rocks. How is a snail going to defend itself? Be really slow. Can't do much. They can make their shells thicker. And in the presence of ocean acidification, the shells are much thinner. It takes a lot more energy to make the shell, and they have to keep battling the dissolution of the shell as the pH goes down. So there's some consequences there. And here's a cool paper. This is actually, these animals were collected in the field. This is a kind of a snail. There's a tremendous diversity. The mollusca is the phylum, and it includes bivalves like mussels. It includes snails. It includes octopus. It's hard to imagine that all those organisms evolved from a common ancestor, but the evidence is pretty strong, including molecular evidence. And this is a snail where you ever think of snails like the one I just showed you, sort of crawling along on its little foot really slowly. Although a lot of snails, the foot has been modified into wings. In fact, they call these sea butterflies, the common name. And so these things, there's the wing up there, and they flap around like this. They have a shell, and they swim around. They spend their whole lives in the plankton swimming. But when you look at the shells, this is the northern part along the coast of California. You see similar things in samples of pteropods taken around the Arctic or Antarctic areas where the pteropods are very common. You can see the shells. This is in naturally collected animals from the field. The shells are dissolving. Okay. So they are clearly perceiving acidification stress already. It turns out there's some effects of ocean acidification I would never have thought to investigate. Here's one. Behavior can be altered. And nobody knows how it's altered, but it's altered in really interesting ways. So reduced seawater, should be one word. Yeah. Reduced seawater pH disrupts resource assessments and decision-making in this hermit crab, Pagurus bernhardus. So here's a hermit crab. Hermit crabs are really weird crustaceans. They don't make their own shells. They occupy shells that are made available when snails die. And so whether or not they have good shells to live in depends on whether snails of the right size die at the right time for them. And there's very little evidence showing that hermit crabs actually ever kill the snails themselves. Here's a hermit crab without its shell. You can see how unprotected the abdomen is. And every time I see this picture, it just makes me want to pick them up by the claws and dip them in melted garlic butter. And I'm sure that other predators see them exactly the same way, maybe without the garlic butter. So a hermit crab without a shell is very vulnerable to predation. And in fact, they will move into anything they can find. So there's a hermit crab that's found up basically a bottle cap to live in. There were no shells. And that's very common in the tropics. People go down in the tropics and they bring back all these beautiful shells. Well, the hermit crabs don't have shells to live in. So you see, so the hermit crabs are living in all kinds of things that are.
On the left, couldn't find a big enough shell.
He's probably in a good shell. If you tap him, he'll probably retract into the shell. Yeah. So we do field sampling with these guys. You take the hermit crab and sort of bang it on the head and see how far back you can pull in the shell as a way of judging whether the shell is the right size, the hermit crab or not. Yeah. It also, ocean acidification impairs the ability of clownfish, Finding Nemo gets into the act again, and the homing ability of this fish. And who would have thought, they would never have gotten, certainly in the U.S. now, you'd never get funding to do a study like that because it seems ridiculous that you'd have an effect of ocean acidification on behavior. So the question is, does reduced pH affect the ability of clownfish larvae to detect their predators from a distance? And it turns, we've known for a while that these larvae can smell a predator from a good distance and swim the other way. So what is the effect of reduced pH, more acidified conditions on that? And there's the larva up there on the right. So the way they did this experiment, you get the clownfish larvae in the lab, and then you raise them at four different CO2 levels. The 390 is the approximate control level that exists now. And then they had 3 other higher levels. And then they conditioned seawater with various predators for 24 hours. And then they test the behavior of the larvae in a flow tank. And you test one larva at a time. And this is the flow tank. So water is coming in tube A, and water is coming in tube B. And there's one fish larva in the tube at a time. And so in tube A, you have control seawater coming in. And in tube B, you have the predator-treated seawater coming in. And every couple of minutes, you check and see whether the fish is swimming. The fish like to swim into a current, so the water's flowing out here. So you want to see whether the fish are swimming into A or B. And they reverse it. So every other time, the control seawater is coming into B, and the predator water is coming into A, just to make sure that the fish don't have a preference for one letter of the alphabet over the other. And they did this every day for 10 days. And every time they used 20 larvae per treatment. They used different larvae every time, so a lot of volunteers for this experiment. And these are the data. So on the y-axis here is the percentage of time that the larva spent in the predator-conditioned seawater. And these are the 4 CO2 concentrations. This is the control one here. And this is kind of interesting. The first few days, about 10% of the larvae are actually swimming towards the predator. They're a little confused. But by day 8, they all get it. And under control conditions, the sensory machinery is all working in all these animals, and none of them are swimming towards the predator-conditioned water. They're all swimming into the control water. But at the higher CO2 levels, by day 4, certainly, about half, almost half the larvae, at least 40% of the larvae, are swimming towards the predator. And at the highest acidity that they tested, the lowest pH, by day 1 is down here, and by the second day, about 80% of the larvae are swimming towards the predator. That can't be a good thing. Don't do it. That's pretty amazing. People also do studies on different developmental stages. And most marine animals have little microscopic larval stages. Again, they're very diverse and often beautiful. And again, hardly anybody in the world has ever seen, because you need a microscope to see them. Snails, worms, mussels, all kinds of bivalves, corals, sea urchins, sea stars, brittle stars, all kinds of crabs, barnacles even, and a lot of fish have larval stages that often look very different from the adult. And basically, you have a fertilized egg, and it turns into a larva, which continues to differentiate, and eventually becomes capable of metamorphosing. And then when it metamorphoses, you get a juvenile. So there are a number of studies that look at the effects of ocean acidification on larval development. So effects of ocean warming and acidification on embryos and non-calcifying larvae of the invasive sea star. So there's the sea star over here, and it's about this big. Right? And there's the larval stage. This is kind of interesting. It's got a 5-pointed radial symmetry, but the larva is bilaterally symmetrical. And it's about a little less than a millimeter. Okay, so again, you need a microscope to see it. Very beautiful little thing, ciliated. And eventually, after several weeks, this thing will metamorphose into that. Okay. And so the acidified conditions, they slow the developmental rate of the larva down. There's much more, like 40% higher mortality, even at low excess CO2 conditions. So people have studied the effects on larval stages of various things too. But there also can be really surprising effects in some studies. And there's something I've been calling latent effects, which is the effects are caused by stresses during embryonic development or larval development. And then the animal metamorphoses. And even if the animal is in a great environment, for some reason, the stress experienced before metamorphosis has a negative impact, usually a negative impact, on how these animals do after they metamorphose. That's called a latent effect. And sometimes, in some cases, you have the stress for a couple days, the stress goes away, the larvae recover completely, and then they metamorphose. And even though the larvae look really fine and nobody has died in the meantime, after they metamorphose, the juveniles just don't do as well. This has surprised a lot of people. So the stress is experienced by the larvae. That's a little snail larva up there, little ciliated swimming organ, metamorphoses, and you see the effect after metamorphosis. And it surprised a lot of people, including me, in fact, because, you think of metamorphosis as a fresh start. So this was, I like this quote, this was actually in the New Yorker a couple years ago. Metamorphosis is a process that is both violent and regenerative, a death and a birth. And you think of the classic example of metamorphosis. Oh, yeah, a new beginning. There was a new beginning. Yeah, love that. So that's a classic example, a caterpillar turning into a butterfly. It's a completely different body plan, a completely different lifestyle, right? Completely different food source. Everything about it is completely different. And it should be, metamorphosis should be a fresh start. But it's not, as it turns out, always a fresh start. And I stumbled into this by accident a number of years ago, and it's become quite interesting. So I'll just show you a couple of examples of some experiments, short examples of that we've done with students at Tufts over the last couple of years. And this is one of my favorite animals. This is Crepidula fornicata. And not only has a great name, it does wonderful things, and it's a great laboratory animal. When these guys start out, this is 1, 2, 3, 4, there are 4 or 5 animals there. When they start out as juveniles, they turn into males. They all become males. And then after a while, they all turn into females. They all go from males to females. And if you put 2 males together, within a couple weeks, the larger one becomes a female and somehow inhibits the other one from changing sex. So if you have 2 juveniles, you always get a mated pair, which is handy for experiments. The other nice thing about them, and so they live in these stacks. They don't have to move around because, unlike most gastropods, most snails, their gills are modified for collecting food particles from the water. So they feed the way oysters and mussels feed. Very unusual for snails. But this whole family, about 100 species, they all do this. And so they don't need to go anywhere to eat. So they form these permanent stacks that are usually attached to rocks or other shells. And they just sort of sit there. It makes it easy for me to collect. You go out at low tide, you bend over, you pick them up, put it in a bucket. You know, you have to get your feet wet. And they release these very nice larvae. So the females, they copulate, and the females brood their embryos for a number of weeks, and then release these larvae into the plankton. And so there's one larva. It's got a shell, it's got a digestive tract, and up this end here, it's got the ciliated swimming organ that's also used for feeding. It also collects food particles, and they feed on phytoplankton. So this one's about 800 microns. Again, you need a microscope to see it, but I guarantee, especially in the early summer, actually through most of the summer, when you swallow a mouthful of seawater, not only are you swallowing a lot of copepods, you're swallowing a lot of larval stages, including Crepidula larvae. No extra charge for that. So they feed on phytoplankton, which are even smaller than the larvae. So these things are maybe 5 microns or 6 microns in diameter. Beautiful, all different diverse shapes, and lots of species. And they all have chlorophyll, so they're all photosynthesized. And even though most people have never heard of them or ever seen them, they are the basis for most marine food chains in the ocean. Copepods, for example, wouldn't be around if they couldn't be feeding on phytoplankton. The larval stages of most animals, including barnacles and snails and echinoderms and sea urchins, things like that. Everybody's feeding on, a lot of them feeding on phytoplankton. Okay. And they also, it turns out, I'm not sure how they come up with these numbers, but apparently the phytoplankton produce about half the oxygen on this planet. That's another reason to be concerned about anything happening to them. And so I have a short video showing, this is one of my snail larvae. If we see this, get this thing work, here's the ciliated organ at the top of the head here. And you'll see in a second, in here, there's the digestive system. You'll be able to see food churning around in the stomach. And over here, you'll be able to see the heart beating. Kind of neat. So this guy is about 600 microns, a little over half a millimeter. There it goes. You can see the heart beating over here, and there's food churning around in the stomach. Yeah. And you can see the little shell. Cute. Make good pets. They're very quiet. Okay. And one female will release 10 to 20,000 of these larvae. Pretty nice. And they release them at about, they're about 450 microns in shell length when they're released. And oyster larvae never get bigger than about 300 microns. So, and these guys, you'll see, they grow really fast too, so they're much easier to work with in the lab than a lot of bivalves are. Most bivalves are. So here's an animal that's metamorphosed, and you can see it's already developing gills over here. This is the digestive tract, and this is actually the intestine the anus actually ends up over the head because during their development, the whole body twists about 180 degrees. How can you not love an animal that dumps on its own head? So, yeah. Anyway, so I'll just quickly show you a couple of experiments we did with some students. There's some work I did at the Friday Harbor Lab off the coast of Washington on ocean acidification. They have a nice ocean acidification system out there. And what we did was we reared the larvae under normal conditions and under low pH conditions, high acidity conditions. And again, these larvae are very tough, so nobody dies. They may grow more slowly, but the mortality is very low. They all eventually get to metamorphosis, and after they metamorphose, either they get to grow up in the high normal pH conditions, or they're subjected to the low pH conditions. So here's a larva. Here's some juvenile. So this is juvenile growth rates, microns per day. You see how fast they grow. You know, oyster larvae, after they metamorphose, or a mussel, maybe they're growing 3 or 4 microns a day. These guys are growing 100 and something microns a day. So this is these juveniles come from larvae that were reared at the normal pH, 8.0, and after they metamorphose, they were allowed to grow on a good diet, and they're growing about 130 microns a day. These guys, the larvae were reared at the 8.0, but after they metamorphosed, the juveniles, poor guys, got to grow at the low pH. You can see the growth rates are much lower, okay, and the shells are much thinner and much less calcified also. But here we have larvae that were reared at a pH of 7.5, so low pH, high acidity, and then after they metamorphosed, their reward was they get transferred to the normal control pH conditions. And so here's the growth rates of those juveniles. And the important thing here is these are growing at about 100 microns a day. These are growing about 140 microns a day. So even though these are now, after they're metamorphosed, they're growing in normal conditions, the experience they had as larvae, for some unknown reason, affects their ability to grow normally under control CO2 conditions. So it's a clear latent effect. Okay? It's something that shows up in the larval development. But things get even better. So here we reared larvae at 2 different phytoplankton concentrations, a high concentration and low concentration, and all the way through metamorphosis. Let them metamorphose, and then the reward for metamorphosing in this study was they get the high food. They all go to high food. And so you see this is now, the larvae are growing at, on the high food diet the first 3 days, a lot faster than they're growing on the low food diet for the first 3 or 4 days. And that was true also if you monitor the growth rate over the first 9 days. The ones on the high food are growing much faster than the ones on low food. They all survive. They all get to metamorphose. And the reward, remember, for metamorphosing is you get to grow at the best, the highest food concentration. And so in both for the first 3 days and the first 6 days, and actually the first about a week and a half, the animals that have been reared as larvae on the low food diet are growing about half as fast on average as the control animals, even though they're all now after metamorphosis on the high food diet. So again, the experience of this low food concentration during early development for some reason influences how well the animals do after they metamorphose. And I'll show you one more example. This is really weird. So here we are growing larvae on 3 different kinds of phytoplankton. So this is one species, one species, one species, and this is the larval growth rate. So on this best diet, they're growing about, in this case, 50 or 60 microns a day. On the other 2 diets, they're growing much more slowly as larvae. But they all survive, they all get to metamorphose, and we then rear the juveniles. And again, in this case, the reward for metamorphosing, it's always something good. The reward for metamorphosing in this case is you get to grow on the best diet as a juvenile. Okay? So here's the larvae on the best diet. Here are the juveniles. They're grown on the best diet from those larvae. They're growing about 100 microns a day. Okay? This is over the first 3 days, the first 5 days. This group is interesting because the larvae were growing much more slowly, but after metamorphosis, it didn't make any difference. The juveniles on the best diet are growing pretty much as fast as these. There's no statistically significant difference in those growth rates. But these juveniles are growing much more slowly. They're being fed as juveniles on the best diet, just like all these others, but they grew up on this diet, and for some unknown reason, the experience of growing up on that diet affects their growth rates after metamorphosis. Okay. what does this have to do with ocean acidification? Well, here's an interesting paper, Global Phytoplankton Decline Over the Past Century. So people are putting records together and looking at how the phytoplankton concentration around the world's oceans has changed over the last 100 years. And it's consistently getting lower and lower. If you have less food, you're going to have problems. And it's not just copepods are going to be growing more slowly. You could have effects even after animals metamorphose. And here's another study that came out recently, ocean acidification-induced food quality deterioration. Oops, food quality deterioration. So it turns out that under increased acidity, the nutritional composition of the phytoplankton is altered, and the food value to the animals that eat them is compromised. That's interesting. So, I guess the question is any of this work, thousands and thousands of studies, is any of this work going to help us predict what the impact of ocean acidification is on the world's oceans? I think you can see why it's probably impossible to predict with any confidence what the impact is going to be, but you can be sure that something is going to happen, and it probably won't be very good. OA is only one of our problems. We have ocean acidification, we have warming waters, we have pollution, we have microplastics coming out. So as I tell my students, it's going to be fascinating to watch, and there'll be a lot of things to document. Species ranges will be changing, and some species will be going extinct. If a prey species goes extinct, then the predator may go extinct, or maybe the predator will shift to start preying on another animal, so they'll be okay, at least for a while. There'll be lots of things to study. But what actually is going to happen, I don't think there's any way of knowing. Actually, we're doing the experiment now. We're all participating. So I guess, you can sit back and just sort of wait and see what happens and study it and report on the consequences, which is it's going to be really interesting. You're going to be hearing a lot more about the ocean over the coming decades. It's going to be fascinating. Or you can write your congressman every week and insist that they, at the very least, honor the 2016 Paris Climate Change Agreement, and also want to do the Science March in Boston on April 22nd. Great, I'll stop there, and I'll take any questions. Thanks.
Thank you, Jen. That was fascinating, but mostly terrifying. So I'll open up for questions now. Okay.
Maybe the biggest worry is going to be depletion of oxygen with the degradation of phytoplankton that contain chlorophyll.
It's certainly, it's a concern. We get about half of our oxygen from the ocean, it turns out. Again, I'm not sure how these numbers are collected, but I've seen this reported a number of times. And so, yeah, that's another concern that you never hear anything about. But the oxygen supply. So maybe, down the road, we'll all be wearing gas masks.
I have a couple.
And I guess if you grow up that way, you won't think twice about it.
I have a couple of questions. The first one, have you seen any transgenerational effects of acidity in your model organism or maybe in other organisms? The second question...
Why don't I answer that one first?
Okay.
Nobody that I know of has, with marine animals, looked for transgenerational effects. There's some work on vertebrates. There's some work on vertebrates, terrestrial vertebrates, showing that under certain circumstances, you do get effects that are passed down across generations. But no one has done anything like that with any marine animal that I know of.
And it relates to my second questions about the reversibility of effects and tipping points. What is the consensus about, what's the limit before everything collapses in terms of pH?
Yeah, I don't think anybody has any idea what the limits are. And one of the issues is that different species are differentially susceptible to the pH. Some species, like crustaceans, seem to be less vulnerable than mollusks, for example, to reduced pH. And we still don't know enough, really, about the species interactions in the ocean. food webs and community structure in the ocean is much, much more complicated than it is on land. Terrestrial systems are simple, very simple by comparison. And of course, on land, you can sort of monitor what's going on. In the ocean, you can't see what the hell's going on. And the larval stages are microscopic, too. So I don't think anybody has any idea, and I don't know if it's possible to have an idea of exactly where things should stop. But it seems like a risk that you don't want to take if you can help it. And it's a problem with living in a democracy where you have to convince people, voters, that this is something we need to worry about, because it's hard to say exactly what's going to happen or when it's going to start happening. But when it does start happening, it's going to be hard to do anything about.
Are there any species that are going to benefit from this? I know in certain areas, certain kinds of jellyfish proliferate and take over.
Yeah, that's interesting. There are growing incidence of jellyfish blooms, and people are starting to talk about maybe we should start putting them on the menu. And in some restaurants, you can actually eat jellyfish now. It may be the food of the future instead of chickens. And instead of chicken, you'll have jellyfish for lunch. Jellyfish seem to be more robust. I don't know if I've seen actually laboratory or field studies on looking at the effects of ocean acidification on jellyfish reproduction or development or survival. Seriously, I'll have to take a look. I'm not aware of any studies like that, but there must be out there somewhere. Yeah, I'm sure some animals are going to be more vulnerable than others, and some are going to be less vulnerable, and some things will go extinct, and that may, at least for a while, the ones that don't go extinct will have less competition, and they may do better. And then we can play off that, and those are the things that we'll start eating.
I have to say that I had a jellyfish last week in Spain, and they're delicious.
See,
So
there you go.
I highly recommend
There's
it.
nothing to worry about, really. Yeah.
I'm thinking about the ocean and the pH in the ocean and the changes in pH and how universal that is, or whether it's worse in the tropics than in the polar regions.
Yeah, I'm not sure. Is the effects of pH? They're certainly not universal. So, in most of these studies that monitor pH, they monitor the pH in the open ocean, and along the coast, pH is much more variable. In intertidal pools, when the tide goes in and out, pH can vary a lot over the course of a few hours, and so intertidal animals maybe are subjected to much more variation in pH than open ocean animals. And they seem to be doing okay.
The dissolution rate between the CO2 in the air and the surface of the water might be slower in colder water.
Right, and I don't know. There's probably literature in chemical oceanography on that topic, on the rates at which the CO2 is dissolving into the ocean.
Well, the dissociation constant
Yeah.
varies with temperature.
Right. Yeah. Yeah, interesting.
You mentioned something about the financial aspects of coral, and I'm just wondering what that is.
Well, I think for corals, the financial benefits of living near a coral reef are tourism. A lot of people go to Australia, the Great Barrier Reef, and other places to snorkel on vacations, and also the reefs provide habitats for a lot of fish species that are fished commercially. Yeah. But if the reefs all bleach, no one's going to spend 2 weeks snorkeling around them, I don't think. So I think tourism is a big problem. Australia is very concerned.
Any final question?
So going along with the coral and the bleaching, I'm just wondering, when that occurs, what happens to that coral? Does it erode eventually, or is it just staying there bleached? And then also, do the populations of fish and other marine animals just move? And eventually the larger issue will be that, there won't be any coral reefs left, or what happens to those animals?
Yeah, well, I'm not sure exactly what, I'm not sure about that literature. When the coral reefs die, the species diversity certainly goes down, because the basis of the food web sort of declines too. When the corals bleach, all you have is a calcium carbonate shell, and so the same thing would happen as happens with empty shells after snails or mussels die. The shells eventually degrade, dissolve, and break apart, and so the same thing will happen to the reef. In the meantime, it would provide still a habitat for fish to hide around in and things to crawl on. So things won't collapse, the whole ecosystem won't collapse completely overnight. But,
All right, we'll leave it at that. Thank you everybody for coming, and
Thanks.
see you next week.
oh, thank you. Thank you. Thanks. Thank you.